Oke, Assalamualaikum warahmatullahi wabarakatuh. Terima kasih sudah teman-teman yang sudah share, yang sudah bergabung di streaming perdana kali ini. Mungkin perlu disampaikan juga bahwa BagIKAN ini merupakan inisiasi udah lama sebenarnya, udah 2 tahun yang lalu. Cuman masih sifatnya offline, jadi teman-teman yang bisa gabung hanya teman-teman yang bisa hadir saat itu. Saat itu masih hanya di kantor. Nah, saat ini saya coba untuk mengaktifkan kembali inisiasi tersebut. Jadi kali ini kita coba yang online. Kali ini saya punya teman lab sebenarnya. Dia namanya MARINE Cusa. Jadi dia mungkin dia akan bercerita banyak. Dia aslinya Prancis, tapi pernah juga di Kanada dan juga pernah di Australia. Jadi kita panggilkan saja langsung bagaimana dia bisa melanglang buana di dunia. Welcoming Marine. Hello Marine, how are you?
I'm very good. How are you, Andika?
Oh, nice to see you. It's very good. It's a good weather actually. So it has been like a week to see the sun, full of sun.
It's been several months that it's been very nice, but this week it's going to get bad again.
Yes, that's true.
Yeah.
So here, MARINE Cusa, she is the science communicator. So you can check his YouTube channel, which is a lot of interesting stuff. I will share all your Facebook link in the comment as well. So,
okay.
And she also the second year of the PhD student in Salford University, working on the traceability using the genetic stuff. And the interesting things, she also the wall climber. So, holding is very interesting. I don't even know how to do climbing. So probably could you tell us a little bit about yourself?
Yeah. Just before, I wanted to let you know, Andika, there's a bit of a problem with the sound. I don't know if it's your microphone, but there's a lot of parasite sound.
Okay.
So maybe it's better if you don't use. I don't know what you can do to change that. Yeah, it's better now, I think.
Okay.
So, yeah, I'm. Well, you did a pretty good introduction already, Andika. So, as you said, I'm actually entering my third year as PhD candidate at the University of Salford, and I don't have a background in genetics. I actually have a background in marine biology. And for my master's, I specialized in Arctic marine biology. So I actually did all of my fieldwork in Svalbard, which is very high north. It's basically at the same latitude as northern Greenland. And I was studying an Arctic fish species. So very different than what you're doing, Andika.
Okay.
Yeah.
So more about the ecology instead of genetics?
I was doing ecology, yes. So I was actually looking at the diet of that species. And I was also looking at the diet of species that are further south, boreal species, they're called.
Okay.
They're not exactly Arctic, they're just a little bit below the Arctic. And the reason why I was doing that is that with the climate change, there's a lot of the species from the south that are going north. Yeah, of course. And so they're kind of encroaching onto the habitat of the species that are living up north. And we're not sure whether or not they're going to be competitors or predators of the species that live up north. And the main question that we had is that up north, really high in the Arctic, you have the polar night. So there's 24-hour night time for months. And the question was, can the species from the south adapt to this lack of light? And can they still see and eat even though there is no light whatsoever? Or are the only species that can actually live in that environment no light six species? Yeah, so it was
it should be like you affect their ability to hunting, isn't it? So they cannot you get used to with the darkness, with the cold as well, isn't it? So it's
so
very
that was the question, and it seems like for a lot of the species that are moving north, like Atlantic cod, it doesn't matter. They don't need light. they still eat even though it's completely pitch black. They're opportunistic feeders, so they just, especially if they're benthic, they just kind of feel what's at the bottom and they just eat whatever is there. And their stomach is full of crap. Sometimes they even eat rocks. It doesn't matter. They just eat everything. Yeah. So the light doesn't matter.
So it's very interesting. Like you said that you have been like, it is also new stuff for you to working with the genetics. It's also remind me that I never touched the genetic before, so this is really new. It's like a nightmare, really, to work with the genetics. Like iPad is really like making you're the closest friend now.
Yeah. It's the thing is, genetics is a little bit fancy, right? And it's kind of it's become really fashionable. Even in the field of ecology, more and more and more people are using genetics, and meta barcoding and this kind of stuff. And so in my mind, it was something that I needed to learn. It was like one of these skills that I should probably acquire, because I don't want to be one of these researchers in the future that sees all of this genetic work and I don't understand it. So it was definitely, I think that's one of the reasons why I decided to head into genetics, and also because I find it fascinating. So, but it is hard. It's hard. It's a very steep learning curve.
So working with the marine stuff is already hard because it is like untouchable, and now you getting into more untouchable thing, working with the genetics, isn't it?
Well, that's the interesting thing is that if you're thinking about environmental DNA, for example, right? Which I don't really work with environmental DNA, but hopefully you'll have Chris on this podcast, and he works with environmental DNA. But environmental DNA is one of these things where organisms swim in the water, in the ocean, and they kind of shed these cells, and then these cells contain their DNA, and that ends up in the water. So you can take a water sample, you can analyze that, you can look at what kind of DNA you have in there, and it can give you information about the species that are around. And so what's interesting is that, as you're saying, studying the ocean is basically studying the invisible, because it's
true.
And you're using the invisible, like DNA, to study the invisible. Like the DNA is going to actually tell you what's there, but you can't see any of that. In the end, it's all just a black computer screen with a list of species.
That's true. That's why I really wonder that even when I do something wrong with it, even doing something wrong with the protocols, I didn't notice when I doing wrong until I got the result when they're in the checking machine. So it's really mystery for me, really, working with the genetics. So the interesting thing is also that you're also working with the seafood traceability. So why the seafood traceability is very important? Because back to Indonesia, we really would like to have to get the MSC certificate for our product, so we can export our product to EU. So what is really matter about the seafood traceability?
Well, I mean, the idea with seafood traceability is that along the supply chain, you're always able to trace back the origin of your product. And so as a consumer, after your product has gone through a whole loads of steps along the supply chain. It ends up to the consumer. As a consumer, you want to be able to trust the information about that product, right? You want to be able to trust that what it says on the package is really what it is.
Yes.
So if it says that this is an Atlantic cod coming from the Barents Sea, you want to know that it's actually an Atlantic cod coming from the Barents Sea. And the reason why that's important is because we know that not everything is sustainable. Not everything that is being commercialized is sustainable, right? Not every fisheries is sustainable. And not every fish that you find on the market is sustainable. So it's up to you. Sometimes it's up to the market as well. It's up to you as a consumer as well to make informed choices. And if you are interested in making these informed choices, you need to make sure that you can trust the label, right?
Yep.
So the idea with traceability is that in the end, it's really, you're basically, for me, what I'm doing is kind of like testing whether or not we can actually trust what's there, what's written there. And so genetics is a very useful tool because you can actually check if the species on the label is actually the right species.
So that's what sometimes we call as the mislabeling sort of.
Yes.
Yeah, because I have heard that sometimes they in some publication they said in the market they sell a product's label as the snapper, but instead of snapper they got from the what you call dory fish. So
yeah, I mean that happens. The substitution of species is a very common phenomenon, and we're not super sure exactly where it happens along the supply chain. It can happen at the level of the fishing vessels, the landing, the processing. It can happen at the retail level, restaurants. So there are many nodes along this supply chain where someone can either mistakenly or purposefully change the species. So if you take the example of, for example, you're a customer going to a restaurant and you want to eat, I don't know, bluefin tuna.
Yes.
And bluefin tuna is an expensive fish.
Yes.
And the restaurant wants to make some sort of margin on that.
Yeah.
Maybe if they're fraudulent, they might replace the bluefin tuna with a fish that is a little bit similar, but that is not nearly as expensive, right? Maybe yellowfin tuna or something. And so as a customer, it's very hard for you to tell the difference. Bluefin tuna with a fish that is a little bit similar, but that is not nearly as expensive, right? Maybe yellowfin tuna or something. And so it's very hard for you to tell the difference. Bluefin tuna with
and some people also have what you call the sensibility of different products of seafood. So sometimes they could be itchy if they eat another fish. So it's also health issues on the mislabeling as well. So
there's health issues, but personally, from what I know of the fisheries world, I think the main issue is really more kind of an environmental issue. It really is. If there are some species, for example, that you're not allowed to fish because they're over harvested, you're not allowed to land them, and fishermen still catch them and land them and then pass them for something else, that's really a problem because in order to understand how much we're fishing, we need to be able to trust what is landed as well. Right? And if the fish are processed on board, and it becomes difficult for regulators to identify what is being landed, you don't know anymore whether or not these, in the case of sharks, for example, whether threatened species are being fished or not. I mean, if you can't identify the parts that are landed, it can really become a problem for the sustainability of these stocks.
So probably we should also greet our friend that has been joined. So they are Seha from Cambodia. So hi Seha. And there's also Abdul Halim. So he's Malaysian. So he's watching, but he would like to go for shopping for Asda first. And there's also Dwi Atminar. So he's also studying about using eDNA in the canal, so he can see the effect of the reservoir to the migration of fish from the downstream to the upstream. So which is very, very interesting, because sometimes we really forget about our freshwater fishes. So we really
know.
Know?
But my name is Marine, so all I care about is just
And there's the question from Afdal. So what we are talking about is supply chain or the product of amphibian. So we working on the fish product, Afdal. So Marine is working on the mainly from the Europe northern species like cod, salmon. So we working on the traceability. She's working on the traceability of those products using the genetics. So we're hoping that by using the genetic, it's help us to tackle all the mislabeling, or what we call it the seafood fraud. So that's what we are thinking now.
And actually, I'm doing two things, or I'm kind of investigating two things. So there's the identification of species using genetic tools is very well established. It's not hard to do, but it is a little bit more difficult to take that to the field. So actually be able to identify species quickly, easily in the field. That's one of the things I'm working on. And then the other thing I'm working on is, can you also use genetics not to identify the species, but to identify where the fish comes from?
Is it able to do that?
Yes. So it's a lot more complicated. It's the same kind of principle. Basically, the more isolated your populations are from each other, so if they are reproductively isolated, the more genetically different they're going to be from each other, right? So if you have some sort of pre-established library, so you went into different regions and you sampled different populations, and then you know which kind of genetic makeup each population has, you should be able to use that to trace back some unknown sample to one of these populations. So it's a little bit like magic, really.
So in other words, you can trace back where the product originally come from, isn't it?
Yeah, but there are some conditions. And for fish, it's particularly difficult because fish move around a lot. Right?
That's very difficult.
There's not a lot of barriers in the ocean. You don't have, huge mountains blocking the movement of species. Fish are highly mobile. A lot of them are very migratory, pelagic. So if there is mixing between populations, it becomes really difficult and perhaps even impossible to use that tool. So it works well for species that are kind of distinct and reproduce in different areas and don't really mix. It can be even more complicated because you can have two populations that reproduce in two different areas, so they are genetically distinct, but then they all come together to feed in the same area.
Okay.
Right? So if your fisherman is fishing in that area, you end up having two populations that have two different genetic makeup. So you have a very good understanding of the ecology of your species before you can use genetics to trace back the individual to a geographical area.
It's really like coming with really basics principle, like garbage in, garbage out. If you know nothing about the ecologies, about the basic thing about your species, and you would like just strike to the fancy stuff using the genetics, it really doesn't mean anything, isn't it?
It's not possible. At least at the beginning, someone needs to do that work. Someone needs to look into the genetics and ecology parts, and then use that to be able to inform, policymakers and people who implement regulations. And it's going to be very much a species by species basis. So for some species it will work. For some species it's not going to work. So it's more difficult than using genetics for identifying species. Using genetics for identifying species doesn't necessarily, it doesn't even matter if you know anything or not about the ecology. It just works. So
that's true. So so for another recent list that's very also interesting. So you also joined the force that helped the UK get away from the COVID-19.
Yeah.
How you end up there, and how your ability, like working in the genetic stuff, now step on into the COVID-19 testing task.
Yeah, it's kind of weird. So there were obviously, to try and tackle this COVID-19 pandemic, one of the things that governments have been doing is to do more testing. And the testing they're doing actually uses qPCR, which is what both you and I are using for our research. And so because that's all I've been doing for the last 2 years, I thought, well, I know qPCR. I don't know anything about human disease, but the method is effectively the same.
Okay.
Yeah, and so I applied, and I was hired. So it's a temporary job. I'm just doing that for a month. So I'm taking a bit of a break in my PhD, and then I'm going to stop in 2 weeks and resume my PhD. So it's let me tell you, it's quite boring. It's very boring.
Oh no.
Yeah. When you and I do genetic work, when we do our research, we conduct the research from beginning to end.
Yes.
We do the sampling, we go in the field, we do the sampling, we extract the DNA,
isn't it?
we prepare the library for sequencing, we do the sequencing, we do the bioinformatic analysis, we do the statistical analysis, we do the writing of the paper, we publish the paper, we do everything.
Yes.
So it's very, it's great. It's multidisciplinary.
Yeah.
Though working in a lab like a COVID-19 lab is very different, because what they're trying to do in a lab like that is to get as many tests as possible analyzed and processed. And so you know how it works, you know how the industry works. In order to be very productive, you need to divide the tasks into small chunks, and you need to have people do only one task repeatedly.
Yeah.
And your samples just go from one station to the next, but the people who work at these stations, they stay at these stations, and they do the same thing over and over and over and over again, right? That's how the industry works.
Yep.
And that's how we get mass production. And that's how we get mass testing.
Yes.
So this is how it works in the COVID-19 lab.
Yeah.
So I'm only appointed to one station, and I do the same thing the same movement all day long.
Okay.
It's very boring.
Yeah, it's very interesting to get the insight from the
It's
expert
really
doing the testing, because we also very in Indonesia we have discussion why we doing so the testing very long, and why this is very take time to from the testing to get the results. So this is the insight. So it's really like a massive sample. So you are only working in the one station. So it's
yeah.
really painful. It's very exhausted for sure, I think.
Yeah.
So we should thanks to all the worker that are working in the testing center really to protect us as well.
Yeah. They're, over the last 2 or 3 weeks, they've hired 300 people to work in that lab, just to work in the one lab where I am. So they really upped their game. Yeah. But one thing is for sure, I like fish better, and I will.
Let's lead to the next question. So Afzal, back to the fish question. So it's mean the study of fish diversity and their genetics. So is it vary from coast to coast? Like the genetic of fish from the coast to coast is different or have diversity?
um. So, are you talking about species diversity, or are you talking about
So is it
population?
possible like one species
Right.
from different coasts is have different diversity in their gene? That I
Yeah.
think the Afzal.
Yeah. Well, that's how it works. Yeah. So from one coast to another, you can have a very different genetic makeup. So if you take the example of cod, I'm taking a northern Atlantic fish because that's what I study. The cod are all around in the North Atlantic. So you have them on the coast of Canada and the United States, and you have them on the coast of Europe, and you have them all the way up until the Arctic. These are all very different populations. They have drastically different genetic makeup, and it's quite easy to trace them back. You can tell if a cod is from the West Atlantic or if it's from the East Atlantic. That's pretty easy to do. But as I said, it's on a species by species basis.
Is it due to the environmental factor that affect their genes, so they have diversity on their own gene?
It can be. It depends what kind of genes you're looking at. So you might be looking at neutral genes that are not really affected by the environment. And in that case, it's just kind of random mutations that happen, and then they're passed on from parents to offspring, and then they end up just being in the population over generations and generations. And these mutations don't necessarily affect anything, but they're unique to that population because they were passed on from parents to offspring for generations. So you have that, and then you do have some mutations, of course, that also occur as a response to environmental variables, or that are selected as a response to environmental variables. So these, and so these might be different from one region to the next as well. Yeah, so you can
probably also lead the factor also to that affected the species diversity, isn't it? Like the environmental factor, place to place is different, so the species diversity also different. Like back to Indonesia, it doesn't have the similar species. Like I never saw the shibas in Indonesia.
Yeah.
What shibas is the Latas calcarifer, which is the white snapper. So it's totally different. Oh, we also have joined the Alex and also my sister and also my brother-in-law. So it is already more than 30 minutes. So I promised to you. So I don't want to butter your your rest time after the working very hard in the testing center.
Oh, I'm not
So-
resting. I'm working on my PhD today.
Oh.
Yeah, I work, you know, when I don't work in the test center, I work on my PhD.
Yeah.
Yeah, that's how it is.
So I would like to probably end up in this session. So if you have time for like or my colleagues to asking, so we are happy for you to post the questions. And if Marian have a chance for he could she could reply to your questions. So
Yeah,
thank you,
absolutely.
Marian, and thank you for all from. It's pretty much for the first time is from around the world. So I got from Australia, Indonesia, and the Cambodia.
Oh,
So
amazing.
hopefully we can also invite people from other the world as well, like Alex. I intend to invite Alex in the future. So he's also the musician. He's also good speaker. So it's very interesting to talk with him.
Nice. I will definitely tune in.
Okay, thank you, Marin.
Thank
Have
you.
a nice day. Bye.
Bye. Thank you, everyone.
Thank you.